Hello dear students, today we will be discussing about the polymorphism in lysosomes. In the previous lecture we discussed about the ultrastructure of the lysosome, and here we will be discussing about the polymorphism in lysosomes. Actually, what polymorphism means: poly means many and morpha means form or structure or shapes. So the lysosome, appears in different shapes, in different forms inside the cells, in different cells, and different stages of the cellular activity. So that's why there is polymorphism in lysosomes. The first, shape of the lysosome, which first form is that's called primary lysosome. Primary lysosome, the first degree lysosome. They are actually small sized sac-like bodies containing inactive digestive enzymes, so are also called storage granules. Storage granules are also called these primary lysosomes or original lysosomes or proto-lysosomes or inactive lysosomes. So simply, primary lysosomes are sac-like structures, small sac-like structures which contain inactive hydrolytic enzymes, and these are actually formed from the Golgi apparatus. When we have studied about the Golgi apparatus, there is cis phase and there is trans phase, and the maturation phase where the finally the which is the trans Golgi, where from these pinching off and the formation of vesicles, which finally certain vesicles form the lysosomes. Those vesicles which pinch off and form lysosome, they are called the primary lysosomes. These are those lysosomes which are just formed from Golgi apparatus, and those lysosomes are actually called primary lysosomes. So primary lysosomes are actually made from Golgi apparatus, and they are just sac-like bag-like structures which contain enzymes in the matrix, and those enzymes are actually inactive. They are not in a position to work. They are just inactive bags of enzymes which are called primary lysosomes. Alright. Then there is second, that's called heterophagosome. Hetero means different and phago means eating, zoom means body. These are also called phagolysosomes or digestive vacuoles. Now, what actually means the primary lysosome changed into heterophagosome, because primary lysosome is inactive. Its enzymes don't work. They don't perform any kind of activity. They are there in an inactive form. Then finally they change into heterophagosome, which is an active bag of enzymes, and they are also, that's why they are called phagolysosome. These are lysosomes which eat something or digestive vacuoles. They digest it. And how they are formed actually: when a food particle or anything which is bigger in size just enters into the cell, and the membrane pinches in and just invaginates and forms an endosome or a phagosome. That endosome, which is food laden, with food laden vesicle which is food laden, or something else, or a bacterial cell or something solid particle, and this endosome will fuse with this primary lysosome. This endosome which has been formed here, this will fuse with the primary lysosome, and finally the primary lysosome will take this endosome inside and just start its digestion. And when this endosome just fuses with the primary lysosomes, its enzymes get activated. Now that's why it becomes a digestive vacuole. Here it was an inactive digestive enzymes. Here the enzymes will become active, and these enzymes will start to digest that endosome which is having that food particle or something else, and perform its digestion. So this is actually a secondary lysosome or second degree lysosome. Then third step is autophagosome. Auto means self, and phagocytosis means that's eating, and then these are actually also called autolysosomes or cytolysosomes, or they are also called autophagic vacuoles. What actually it means that sometimes when we are facing starvation, there is no food available to the cells, and during fasting or starvation, or when there our certain organelles are growing old and they are about to die. In such situation or starvations or certain pathological conditions, these lysosomes just surround these uh, organelles. For example, there is mitochondria, there is fragments of endoplasmic reticulum, and these surround them and just take them inside and just break them down and just digest these organelles to draw energy from these energy. This is done because of to renew, rejuvenation or renew to renew the cell, to rejuvenate the cell. So to get the energy from these uh, organelles like mitochondria and certain fragments of endoplasmic reticulum. So it's actually doing the self digestion of its own organelles. That's why it's called autophagosome. So autophagosome is actually called autolysome or cytolysosome or autophagic vacuole. Then finally, we have residual bodies. Residual bodies, this is the final. This is also called a telolysome or tertiary lysome. These contain the undigested food like the fats, glycogen, or cellular debris. They are finally expelled out of the cell. Residual bodies are actually, when cell performs its digestion. For example, it has taken the food particle, it has taken the bacterial cell, and when it finally completes the digestion, some things remains there that's left off. For example, when we eat food and our stomach and intestines perform the digestion, and some sort of food is there which is left undigested and just thrown out. Likewise, inside these lysosomes, certain things of the cellular debris or the food particles which are here, which are not digested fully. That, for example, fats, for glycogen, and they just remain in that uh, lysosome, which forms a residual body, and this is then thrown out of the cell or exocytosis. This is how the lysosome comes in different forms, showing polymorphism, having different activities at different stages of